Hello, good morning all of you dear students. Today we are going to listen about a very important topic called biophysical techniques, and the innovation of this today's topic is entire topic will be dealt with students, and here this 4 of the students coming from P.R. Government College Kakinada, and she is Usarani and Regina studying biotech second year, and Rupa Devi and Prasannata studying final year BHC, and I thank very much our Commissioner Madam for conceptualizing and designing this program, that is a student as teacher program. Today very important biotechnology principles, the procedures and applications they are going to dealt basically from chromatography onwards up to what you call electrophoresis and spectrometry, spectroscopy, whatnot. Many physiologists, biochemists, microbiologists, virologists, immunologists all use. Without these techniques even the genetic engineering cannot be so much progressed. So, with this introduction you will enjoy the experiential learning of these students, and I also request the participatory method of from the students who are watching from different colleges, and the last 10 minutes will be for the student interaction. So, raise your queries or questions, they will definitely answer with confidently. And now I request the first student to present the biophysical techniques. Thank you.
Good morning everyone, this is K. Usharani. Today I would like to explain you about biophysical techniques. The techniques which are used to separate biomolecules like carbohydrates and amino acids basing on their physical properties like size, charge, color, etc. of those molecules is called biophysical techniques. Some of the techniques which we are going to deal today are chromatography, electrophoresis, spectrophotometry and centrifugation. Now I would like to explain you about chromatography. Chromatography is based on a difference in rate at which components of mixture move through a porous medium, that is stationary phase, under the influence of solvents or gas, that is mobile phase. This chromatography is differentiated into two kinds, that is partition chromatography and adsorption chromatography. In partition chromatography, it distributes between two immiscible phases, while coming to adsorption chromatography, it adsorbs on a stationary phase. The best example for partition chromatography is paper chromatography, and adsorption chromatography is again differentiated into two kinds, that is thin layer chromatography, column chromatography. Based on stationary phase, this column chromatography is again differentiated into three kinds, that is gel permeation chromatography, ion exchange chromatography and affinity chromatography. Now I would like to explain you about paper chromatography. Paper chromatography mainly works on the principle of partition or distribution coefficient, which describes the way in which a compound describes itself between two miscible phases, that is stationary phase and mobile phase. There are two phases in paper chromatography, stationary phase. Mainly Whatman number 1 filter paper is used as a stationary phase. While coming to mobile phase, solvents like for example for amino acids, N-butanol, acetic acid and water are used as solvents. After selecting the stationary phase and mobile phase, then comes to the procedure. The procedure of this is taken down by 4 steps, that is choice of filter paper, sample application, choice of solvent system and paper development. Choice of filter paper. Mainly Whatman number 1 filter paper is selected and it is cut into required dimensions. Once the paper is selected, it has to be washed with 0.1 normal of HCl to remove impurities. After removing impurities, a line from the base of 2.5 centimeters is drawn, and after this sample is applied. The sample application is based on 2 steps, that is mainly sterilized toothpick is used for sample application or capillary tube. The sample application should be like this and with a distance of 1.5 centimeters from each sample. And after this the paper or the sample should be kept in the paper should be kept in suitable solvent and the paper is developed. After paper development, paper based on separation paper development is of 4 types, that is ascending paper chromatography, descending paper chromatography, 2-dimensional paper chromatography and radial paper chromatography. In ascending paper chromatography, the paper is ascended from downwards and the only force acts on this ascending paper chromatography is capillary force. Due to this capillary force, the molecules move very slower and the accuracy is very perfect. While coming to descending paper chromatography, it is descended from the above of the chamber and 2 forces acts on it, that is capillary force and gravitational force. Because of this gravitational force, the molecules move very faster when comparing to ascending paper chromatography. For this reason, the ascending paper chromatography is obviously used, and descending two-dimensional paper chromatography. In two-dimensional paper chromatography, two solvents are usually used. The components which cannot be separated using single solvent, in such cases two-dimensional paper chromatography is used. After completion of paper chromatography, the sample paper is kept in hot oven air and solvent front is marked. After marking the solvent front, if the sample is colored, then no necessary of using any visualizing agents. If the sample is colorless, then visualizing agents are used. For example, for amino acids, ninhydrin is used, which gives purple color when reacted. And after this, it is identified by using RF factor. RF factor is retardation factor. Retardation factor is distance moved by the solute origin to the distance moved by the solvent front. This is identification of this is a diagram showing identification which shows purple color, and it is applied in separation of molecules and also in the separation of ripening and fermentation. Now, I would like to explain you about column chromatography, which is the best example for adsorption chromatography. It works on the principle of mixture of compounds dissolved in mobile phase is introduced into the column. The individual components differentiates depending upon their relative affinities. It is a principle of column chromatography, and this column chromatography is similar to gel permeation chromatography, ion exchange chromatography and affinity chromatography. This column chromatography also has two phases. One is stationary phase, another one is mobile phase. The ideal properties which a stationary phase should have is, it should be uniform in size, spherical in shape, free flow of ions, and it is inexpensive. Mobile phase. The choice of solvent naturally depends on solubility of substance. It has three functions to perform: introduction of sample, development of chromatogram and elution. Once the mobile phase and stationary phase are selected, then the column is packed. There are two methods of column packing, that is dry packing method and wet packing method. In dry packing method, the adsorbent is directly taken into the column. While coming to wet packing method, the adsorbent is made into a slurry with an organic solvent and then introduced into the column. After column packing, these are eluted out. There are two types of elutions, that is isocratic elution technique and gradient or stepwise elution technique. In isocratic elution technique, single solvent is used as elutant. While coming to gradient or stepwise elution technique, the pH, ionic strength and polarities of the elution is changed gradually. Now, there is a small video on column chromatography, watch it. After this, my friend is going to explain you about the rest of the techniques. Thank you. Then this is about column chromatography. This is the present is the column showing, and after this add enough solvent, usually lower polar solvent. Then tap the column, pour silica and slightly stir the silica. Slowly introduce into the column, and after introducing the mobile phase, it is a mixture of components, mobile phase, and after that we have to rinse the column with the silica, because no impurities must be present in the column. That is a absorbent of silica showing you, and mark the silica level and gently tap the cork rings or hand soft materials. By using soft materials, we have to tap the column. Here we are using two rubber corks to tap the column. Tapping should be very smooth and we have to rotate while tapping to adjust itself with the column. After tapping, a small amount of pressure can be applied. Now we have to load the sample loading. We have to dissolve this is the mostly used packing is wet packing. We have to dissolve the sample with an organic solvent and then pour into the column. Sample adsorbed to the silica after rotary model. Pour dry sample and rinse the wall after pouring the sample, because no sample should be in the walls of the column. This is a sample showing you. Now we have to put sand on the top of the sample, because no impurities must go into the column. Care must be taken while doing all this process. Then the elution is collected out in fractions. This is a video of column chromatography and rest will be explained by my friend Rizina. Thank you.
Hi friends, this is Rizina. Today I am going to explain two techniques which comes under column chromatography. The first technique is gel permeation chromatography. It is also called as gel filtration chromatography, and it has a principle to work on, that is the separation of samples is based on their shape but in some cases by their molecular weights. This is the commercial column which we use in a gel permeation chromatography. This diagram represents a column with a solution and with a gel material. The blue crescentrix resembles the gel material with porous nature. The blue and the red and purple ones resembles the molecules which are present in the column. The purple ones are smaller particles can enter into the gel and elute lately. The red ones are the large particles which cannot enter into the gel material and elute sooner. Here gel acts as a stationary phase. The gel should be chemically inert and should have a high mechanical rigidity and should have a uniform particle and pore size. Commonly used gels are dextrons, agarose and polystyrene. For the separation of proteins and biomolecules, we use Safrax gels. Basing on the samples, Safrax gels are different types. Here I am representing the types of Safrax gels, that is Z25, Z50 and Z75, Z100 and Z200. Before going to the process, we have to prepare the column. For the preparation of column, we must do the gel material into the swollen form by adding a proper solvent. After making gel material into swollen form, we have to incubate the gel material into the column in the form of slurry. After all this process, we have to add a sample on the top of the stationary phase. After the adding of sample, we have to add a buffer for elution. Here elution is isocratic elution, and it is the diagrammatic representation of particles how they eluted. The first peak resembles the larger molecules elute sooner. The remaining are the different molecules which elute later. After all this process, we have to collect the fractions from the column and examine under the spectroscopy so that we can calculate the concentration of polluted particles. It is a small video which represents as gel filtration chromatography.
In gel filtration chromatography is based on the differences in sizes from biomolecules as they pass through a column packed.
It is gel which is the applications of gel permeation chromatography has different various types of applications. That is, first one is separation of lower molecular weights, and the third one is we can determine the molecular weights of macromolecules. The next technique is ion exchange chromatography. it is a process that allows the separation of molecules, and polar molecules is based on their affinity to the ion exchanger. Ion exchangers are different, two types. The first one is cation exchanger and second one is anion exchanger. Here is the diagrammatic representation of anion exchanger and cation exchanger. We use resins as a stationary phase here. Resins are amorphous particles of organic metals. We use polystyrene resin as a stationary phase in ion exchanger chromatography. Resins are made up of copolymerization styrene and divinyl benzene. Here it is the diagram which shows the structure of polystyrene. Basing on the samples, ion exchanger resins are 4 types, that is a strongly acidic cation exchanger, weakly acidic cation exchanger, and strongly basic anion exchanger and weakly basic anion exchanger. Before going to the process, we have to prepare the column. For the preparation of column is same as we prepared in the column chromatography, but except 3 points we should observe here. The first one is we have to pre-cycling the medium or swelling the medium. By making the pre-cycling the medium, we can make a burial functional groups will exposed, and the second one is removal of very fine particles which are present in the exchanger, and the third one is exchanger will have to be equilibrated. Once a column is prepared, we have to transfer the sample on the stationary phase. After adding a sample on the stationary phase, we use buffers for elution. For 2 types of ion exchangers, we use 2 types of buffers. For anion exchanger, we use cationic buffers, that is tris and pyridine. For cationic exchanger, we use anionic buffers which are called acetate and phosphate. After adding a buffer solution for elution, elution occurs. Elution in ion exchanger chromatography is gradient type. After all the elution occurs, we have to collect the fractions from the sample. This is the diagrammatic representation of a column with the resins which are filled on the column. The sample we applied on the top. The gel bits resembles the positively charged which are present in the resin, attracts negatively charged which are present in the sample. This is a binding column with a positively and negatively charged molecules. After binding, we have to add a elution buffer for elution. After elution, fractions are collected in a test tubes. This is the video on ion exchanger chromatography.
Upon the reversible adsorption of charged solute molecules to immobilized groups of opposite charge.
These are
Most
positive
ion exchange experiments are performed
resins
in
we
4
use
main stages:
as a stationary phase.
equilibration,
And
sample application and wash, elution,
we have the equilibration and sample
regeneration.
application and wash, elution, regeneration. These are resins which are present in ion exchangers. Inside the resins, we can see that the positively charged attracts negatively charged, and it is equilibration occurs. After equilibration, sample application is done. The negatively charged which are present in the sample are replaced by the negatively charged one which are present in the solution. Hence, it is the graphical representation of a elution. Elution occurs and replaced by negatively charged ones.
The number of charged groups on their surface. The final step, regeneration, removes all molecules still bound. This ensures that the full capacity of the stationary phase is available for the next run.
Here we can see the applications of ion exchanger, that is, we can determine the composition of nucleic acids, and is also useful for the separation of many vitamins, other biological amines and organic acids and bases. Thank you one and all.
Hello friends, this is Sri Rupa Devi. Today I would like to discuss some few points about electrophoresis technique. Coming to electrophoresis. Electrophoresis is a migration of a charged particles or the molecules in a solution under the influence of an applied electrical field. In general, naturally obtaining material or the biomolecules such as amino acids, peptides, proteins and the nucleic acids which possess ionizable groups and therefore separated and analyzed by using this type of electrophoretic techniques. Coming to the basic principle of electrophoresis. The basic principle of the electrophoresis is a charge by mass ratio. The molecules which have similar charge will possess different charge by mass ratios due to the difference in molecular weight. This difference in the molecular weight will lead to differential migration of an ions in a solution when subjected to electrical field, and the electrophoretic mobility mainly depends upon the sample, applied electrical field, ionic strength and the buffer composition and the ionic strength. And these are the types of the electrophoresis. It is majorly classified into two types. First one is free electrophoresis. It is the electrophoresis without the stabilizing medium, and the second one is zone electrophoresis, the electrophoresis with the stabilizing medium. Free electrophoresis is again classified into two types. First one is microelectrophoresis and the second one is moving boundary electrophoresis. And coming to zone electrophoresis, it is classified into two types. First one is paper electrophoresis and the second one is gel electrophoresis. This gel electrophoresis is mainly used nowadays, and the basic gels which we are used nowadays are starch, agarose and polyacrylamide. And this is the gel electrophoresis. The separation of the gels not only depends upon the charge of the molecule but also on the size, whether the molecule will enter the pore and will retard or will bypass it. And these are the types of the gel electrophoresis. It is of two types. First one is horizontal gel electrophoresis and the second one is vertical gel electrophoresis. Agarose gel electrophoresis is example for horizontal gel electrophoresis, and SDS-PAGE is example of vertical gel electrophoresis. And agarose gel electrophoresis. Agarose will form a gel or a molecular sieve that supports the movement of small molecules which are used for DNA. And this type of agarose gel electrophoresis is mainly used in the analysis of the DNA. And it is made up of a linear polysaccharide units with a molecular weight of 12,000 kilodaltons. And the gels are prepared at 0.8% to 2%, providing tunnels for the molecules to move through the gel. And this is the diagrammatic representation of the agarose gel electrophoresis. Before going into the instrumentation, I would like to explain the components of the agarose gel electrophoresis. These are the boards which are used to cast the gel. And these are the combs which helps us to provide the wells in between the gel. And this is a gel casting unit which is used to cast the gel. These are the boards which are placed in the gel casting unit. And this is the electric power pack which we use to supply the electric field. And this is the electrophoretic chamber in which the entire electrophoresis will take place. After the explanation of the components, let us go to the instrumentation. For the instrumentation, we have to prepare the sample. For the preparation of the sample, we have to weigh the required amount of the agarose and add it to the buffer solution and dissolve it. And after that, we have to place the gel in the gel casting unit, and after that, we have to apply the electric field. After the applying of the electric field, the migration of the ions from one end to other end will take place, and the separation of the molecules will take place mainly upon the molecular weight of the DNA fragments. After the separation is completed, the gel is placed under the UV trans illuminator so that we can observe the bands of the DNA. And coming to SDS-PAGE. SDS-PAGE means sodium dodecyl sulfate polyacrylamide gel electrophoresis. It is generally used to separate the proteins. The main use of the SDS-PAGE is it acts as an anionic detergent and it helps in denaturation and unfolds the proteins, and it gives the negative charge to amino acids present in the protein sample. And the preparation of the gel. Gels can be made at different concentrations of acrylamide and bisacrylamide. For example, the gels made at 3%, 6%, 9% and 12% will produce different openings for molecules to migrate. The larger the opening will allow the molecules to move through the gel. And coming to sample preparation, the protein sample usually dissolves, that means it has to boil for 15 minutes in a tris buffer. This is a basic buffer which we used in the electrophoresis, and its pH should be maintained at 6 to 8. And the second thing is SDS. As I have already told in the previous slide that SDS means sodium dodecyl sulfate. And the third one is beta mercaptoethanol. And the next one is a tracking dye. The most commonly used dye in this sample preparation is bromophenol blue. And all these components should be mixed along with the sample. And after that, gel packing. Coming to gel packing, it is of two types. First one is stacking gel and the second one is separating gel. The main use of the stacking gel is it stacks the entire proteins present in the sample. After the protein stacks, it enters into separating gel where the actual separation will occur. And here also the separation is based on the molecular weight of the proteins. And this is the diagrammatic representation of SDS-PAGE. This is the instrument used for SDS-PAGE. This is the upper reservoir and this is the lower reservoir. In between the upper and the lower reservoirs, we have to place the gel. And these are the components. This is a gel casting unit which we cast the gel. And these are the spacers which are used to provide the gaps in between the gel plates. And these are the combs which are used to provide the wells in the gel. And for the apparatus, and after the preparation of the sample, we have to place the gel plates in the electrophoretic chamber. And here also the separation is based on the molecular weight of the proteins. And the last one is the detection. Different types of dyes are used for detecting different type of compounds. For example, if we want to detect the nucleic acids like DNA and RNA, we use ethidium bromide. And for proteins, we use Komatsu brilliant blue as a dye. Thank you for giving me this opportunity, and the rest of the techniques will be explained by my friend B. Prasannatha.
Hi, hello my dear friends. A pleasant good morning to everyone. This is B. Prasannatha studying BSc Final Year. Today I would like to deal with an instrument that is spectroscopy or spectrophotometer. Before going to my topic, that is spectrophotometer, now I would like to explain what is spectroscopy or spectrophotometer. It is the study of interaction of light with matter is known as spectrophotometer. Suppose if you imagine two samples of the same substance, the darker one is more concentrated than the lighter